Dear audience, first of all, I would like to thank for the opportunity to give a presentation today. I'm Inez Bosnyák, a sixth-year medical student and MD PhD student from the Department of Anatomy, University of Pécs Medical School. In the following minutes, I'm going to talk about an optimal ischemic retinopathy mouse model and the consequences of oxygen deficiency in the retina. First, I will give you a brief introduction about the retinal layers. The retina is the internal layer of the eye, which consists of several layers. The axons of the ganglion cells are in the nerve fiber layer, while the cell bodies of ganglion cells are in the ganglion cell layer. The retinal ganglion cells are neurons in the periphery, which have a very important role in the vision. Then the cell bodies of bipolar cells are found in the inner nuclear layer, while the cell bodies of photoreceptor cells are in the outer nuclear layer. And between the nuclear layers, we can find plexiform layers where the synapses are found. Besides this, I would like to highlight the photoreceptor layer where the rods and cones are found, and the pigment epithelium has also a very important role in the light transmission. Now let's see a couple of words about the circulation of the retina. The internal layers of the retina are supplied by the central retinal artery, which comes from the ophthalmic artery, which is a branch of the internal carotid artery. The internal carotid artery is a branch of the common carotid artery. The common carotid artery will be very important for us, so we will talk about it later. However, it's not so easy. So you can see the capillaries from the central retinal artery here. However, in the outer retinal layers, we have no blood vessels. So the outer retinal layers are supplied by the so-called choroidal vessels. So in summary, the retina has very high oxygen consumption and metabolic activity, so lack of oxygen supply can cause visual impairment. Retinal hypoxia has a key role in the pathomechanism of the diabetic retinopathy, age-related macular degeneration, glaucoma, and the retinopathy of prematurity. Based on the literature, the damage of the inner retinal layers are more severe. However, many contradictory data are found, for example, in case of the ganglion cell damage. Despite the prevalence and the severity of diseases, we have no effective side effect-free treatment. That's why our aim was to develop an optimal ischemic retinopathy mouse model to investigate the cell damage in time-dependent manner to find the most sensitive cell types. Besides this, we were curious about the ganglion cell damage. However, you may ask that what is a biological model and why do we need biological models? Let me show you an example. You can see the arteries and nerves of head and neck region in this picture. This is a human skull, and the yellow structures are the nerves, while the red structures are the arteries. You can see how difficult it is. And if we would like to analyze the structures one by one, we can build a model for that. So we can study the external carotid artery with this model. So that's why our aim was to develop an ischemic retinopathy mouse model. However, it was much more difficult than we previously expected because those models we could find in the literature were not appropriate for our aims. Now let's talk about our methods. Retinal ischemia was induced by bilateral or unilateral common carotid artery occlusion. So we made a small incision in the middle of the neck, then we isolated the common carotid arteries on one side or both sides. And then we occluded the arteries with surgical microclips or with sutures. So we performed BCCO for 10, 13, 15, or 20 minutes. Besides this, permanent unilateral common carotid occlusion was also performed. And the thickness of retinal layers were investigated by optical coherence tomography. OCT is a painless in vivo method, which is also widely used in the clinical practice. This is a picture about an OCT measurement in our laboratory. And this is a representative picture. In the middle, you can see the optic nerve, and we can identify all retinal layers one by one. And after the measurements, the software analyzed the layers, and we get their thickness in micrometer. So OCT measurements were performed 3, 7, 14, 21, and 28 days after the surgery. And after the measurements, a complex multi-step statistical analysis were performed, and the figures and the analysis were made with R programming language. Besides these, molecular methods were also performed, so we investigated the number and distribution of ganglion cells and the vascular density on whole mount retina preparations. And we were curious about the photoreceptor damage too. So in the following video, you will see the retina preparation process. So this is an isolated, fixed whole mount mouse retina, which is already labeled by primary and secondary antibodies. This video is speeded up a bit because the retina is very sensitive tissue, so it's very hard to manipulate it without tissue damage. And after this preparation process, we mounted the retina with fluoroshield, and the results were analyzed under fluorescent light microscope. Now let's see our results, begin with the OCT ones. In the experimental period, more than 400 OCT measurements were performed, so we got almost 5,000 data. This graph shows the total retinal thickness. The first column is the 20 minutes BCCAO group, then 13, 15, and 20 minutes. The vertical axis represents the days after the surgery. And these horizontal lines are the so-called confidence intervals. So we can talk about significant change if this line does not cross this vertical one. So as you can see, we did not experience significant change in the total retina. However, the change was almost significant in case of the 20 minutes groups. So you can see the middle layers of this picture, and the middle layers significantly changed 7 and 14 days after the surgery in the 20 minutes group compared to controls. And similar results were found in case of the inner nuclear layer and in case of the outer nuclear layer as well. So 7, 14, and 21 days after the surgery, we experienced significant change in the outer nuclear layer in case of the 20 minutes groups. Besides this, we were curious about, for example, the photoreceptor damage as well. So immunohistochemical analyses were also performed. The first picture is the control group, then 20 minutes BCCAO, then UCCAO group. The green color shows the cones along the whole picture, while the red color shows the rods, and you can see all cell bodies with the blue color. So we experienced cone loss in case of the 20 minutes group, as you can see in the pictures here, and this was much more remarkable in the case of the UCCAO group. So we had almost no cones 4 weeks after the surgery in the UCCAO group. And besides this, we were curious about the ganglion cell damage as well, so we evaluated the number of ganglion cells in the central and peripheral regions. The left side is the control group, the right side is the UCCAO group. So these small red dots are the retinal ganglion cells, and you can see by naked eye the decrease in their numbers. However, after the quantification, we experienced significant decrease in the ganglion cell number in case of the 15 and 20 minutes BCCAO groups and UCCAO group as well. Besides this, we analyzed the vascular density. So you can see a representative lectin-stained whole mount retina. So the vascular density increased more than 20% in case of the UCCAO and 20 minutes BCCAO groups, which means that new vessels were formed after the ischemia. So in conclusion, the 20 minutes BCCAO model is good for investigating the consequences in ischemia and reperfusion in a time-dependent manner and to find the most sensitive cell types. And besides this, the UCCAO group is good for testing potential new drugs because we can cause more severe damage in a short time. Besides this, I would like to highlight that the small changes with OCT measurements can cause by very severe tissue damage, so the small OCT changes must be taken very seriously in order to treat these diseases effectively. And finally, I would like to thank you for my mentors, Professor Dóra Reglődi, Dr. Alexandra Váci, and I would like to thank you for all members of our retina research group. Thank you for your attention.